And you can change the slides, right?
Yes, I have that power.
You have power. Okay.
Okay, I'll be forever grateful to Adam for that last slide because I was really struggling for a segue. A segue into terrestrial mammal bioacoustics. And so I'm Lisa, and I would not define myself as a bioacoustician, but rather an evolutionary biologist with some interest in disease ecology, and recently, thanks to CSearch and the team that's here today, introduced to this world of bioacoustics. And so I'm very grateful to you guys as well for sharing your knowledge and for the opportunity to be here. Just like Catherine, I did all of my primary learning at UCT, so I'm originally from around here. But I, quite a number of years ago, made the big leap to the savannas of northern South Africa, where I live now in Kruger Park, and I work as a research coordinator for Yale University there. And so I have the great fortune to have access to some wildlife to record. And so together with CSearch and SDU, we've been exploring many of the ideas that you've heard about today, about doing some of these ABR measurements in megaherbivores. But I'm just going to talk about two really exciting projects that have come out of this kind of collaboration with everybody. And one of those is using acoustics to study disease, which aligns very nicely with my current job, where I am studying bovine tuberculosis in African buffaloes in Kruger. And the second, a little bit more along the lines of what you've spoken about today, not acoustic localization in wild hippos, so it's not the hippos that are doing it, but we are listening and trying to use sound to find the hippos, if that makes sense. So I'll start with this first one. If this term acoustic epidemiology is new to you, that's good, because it was new to me as well. And it's basically this very new field that uses body sound data to monitor disease. And obviously it launched in humans, and you can see this cough monitor that this is out of a scientific paper really, this person is wearing. And you can use this information to study the sounds produced by the body, such as breathing and coughing, and this is often to do with monitoring health outcomes. So this graph over here, I'm sorry if you can't see it at the back, but it shows cough episodes per hour and treatment day for somebody with a respiratory disease. And you can see that the cough sounds reduce as disease, as treatment progresses. And so Adam spoke a little bit about technological advancement and the miniaturization of devices, and I'd like to layer on top of that the use of artificial intelligence in helping us to analyze these massive amounts of data that I'm sure you guys know come from audio recordings, and it can be quite difficult to manage. And so the goal is to use sounds as biomarkers of disease screening, and it's started in humans. In fact, during the COVID-19 pandemic, this was a paper that came out where they launched an app and you could cough into the app, and with some degree of accuracy, an artificial intelligence-enabled platform told you whether or not you had COVID. Wow. They gave it a shot, right? And so that's quickly moving through what's happening in human health. Where this has really come to the fore in animals is precision livestock farming. And this is very advanced technology deployed on farms to monitor the health of livestock, right? And so the sounds that those livestock are making tells you something about their welfare, which I'm not going to talk about today, but there's a whole element of the study to do with welfare, but also about their health. So they've utilized bioacoustics in livestock with the goal of increasing productivity and making more money, but at least the data are there. This is a review from this year, talking about whether or not we can reliably detect respiratory diseases in farm animals. And a lot of the basis for what I'm going to talk about comes a little bit from that paper as well. So that's the background, and I think you can see where we're going with it. So I'll tell you a little bit about BTB, bovine tuberculosis, caused by a bacterium called Bacterium bovis. It's a zoonotic disease. That means it's easily transmitted to other wildlife species. It's zoonotic for humans as well, so we have to be very careful around animals that have contracted this disease. Of course, once it's in a wild population in Kruger National Park, it's very hard to treat or control this disease. But it's there, and it is endemic in Kruger National Park. And there's persistent concern that there's going to be disease spillover from the animals inside the park to the cattle outside the park. And so there are numerous studies that have been launched to better understand the dynamics of tuberculosis under this umbrella of One Health. Because once you get bovine tuberculosis into your herd of cattle, the current best practice is test and slaughter. And that costs people a lot of money, and communities don't like it, and they lose their livelihoods, and it's really terrible. So we often, once it spills outside the park, we have to quarantine those animals, and obviously affect people. So you have a question?
Yeah, it's a lion. Do they get tuberculosis too? Or?
Wait.
Sorry.
Yes, I'm going to talk about the different species that it affects, but this kudu over here, you can see these big lumps that form. That's diagnostics of TB. And then in predators, they get very swollen joints and obviously massive loss of body condition when they get sick. And the reason, not working.
We need to do something.
I don't know what's going on.
I spoke for too long.
No, need to go here. I think it's good to do that.
No? I'm too scared to try any other buttons. It's working over there.
That's weird. Try it again. Okay. Or I can.
Working on a small
The thing is, oh, I know now. Just a second.
Okay. Are there any questions so far?
It's a picture of the lion. Is dead or just that you studied him?
This animal was alive, but they darted it in order to euthanize it. For two reasons. I mean, obviously because it's suffering, but also because it represents a source of disease that it could spread.
Okay, I think you're on now.
There we go.
When was this, you said it's endemic to Kruger, right? When was it discovered?
You're less patient than Jakob, it's coming.
That's already something.
So these lumps in the animal, they form because your body is trying to, this happens in human tuberculosis as well, your body is trying to contain the bacteria, and it does that by forming granulomas, these big cysts that encase the bacteria and try to stop it spreading. Yes?
And that's got nothing to do with like lymph nodes or whatever, it just forms a ball, like
No.
it forms almost like a tumor.
No, these are the lymph nodes.
Okay.
These are the cross-sections of the different lymph nodes, not of this kudu, but of a bushbuck. So the lymph nodes get incredibly swollen. The granulomas primarily affect the lymph nodes and the lungs.
Okay.
And so you get these macroscopic granulomas, and you also get microscopic lesions in all of the animal's tissues, wherever the bacteria. Yes. Okay. Oh no. Hey.
That's me. That was me.
Yeah, I know.
Sorry.
Okay, we're good.
But you're not in sync with the screen, which bothers me.
That's a bit strange.
Yeah.
Oh, that's probably going to confuse Catherine.
She can get the.
Okay, so very briefly, bovine TB is not from Africa, though there is an African clade, you just don't get it here. This was introduced in the late 1880s and first recorded in wildlife about 100 years ago. And it was slowly discovered in many different wildlife species. So I've listed a few here. lions, kudu, warthogs, leopards, everything from a mongoose to an elephant. They found bovine tuberculosis. So it affects lots and lots of different species. I've mentioned that other stuff. And what's interesting is we know quite a lot about bovine tuberculosis in cattle. So that's this map on the right, absence in white, clinical signs in black. So there is bovine tuberculosis recorded in populations, but we know almost nothing about its dynamics in wildlife. So opening up an easy way to test for it would be a cheap way to test for it, be a really important step in diagnosis. Hey. Okay, so then we come finally to the buffaloes. African buffaloes are a reservoir host for bovine tuberculosis. That means that many buffalo have the disease, but few of them show clinical signs, even when they're shedding bacteria. So they shed the bacteria while they're feeding, while they're drinking, and then other animals forage in the same patch or drink the same water. They contract the bacteria and they get sick and die. So, yes?
How is it shed when they drink? Like through saliva?
Yes.
Okay.
The first introduction of the disease was in the south of the park, and it's been monitored carefully for the last 50 years or so, and it's been slowly progressing through buffalo herds from the south to the north. Obviously, it's impractical to cull thousands and thousands of buffalo, so that's not an option. And managing disease in wildlife populations is very expensive, and just testing the animals is super expensive. So first of all, you have to immobilize them to get the tests, which we spoke about. Smaller animals, much simpler, but with the buffalo, in a big 5 area where you have dangerous animals around, you have to be very careful about that. There's the expense of the number of staff that you need to manage the situation, keeping a helicopter in the sky, the crushing expense of the immobilization drugs, because you need an awful lot of ketamine or thionyl or azapirin to immobilize a 500 kilogram animal. So it gets very expensive, but there are active studies like the one that I work for that are testing and studying TB in the buffalo at this great expense, right? Interestingly, this last study that we did included electrocardiograms of buffalo to monitor their heart rate during immobilization, and that, I don't know, so it made the ABR measurements feel a little bit easier to me, because we have electrodes on that animal already. And the collaboration necessary to study this is also big. So this is Lynn Marie, she's the state veterinarian. We've got vets from the University of Pretoria and Sandbox. The number of people you need to do this kind of monitoring. My point is, it's cumbersome. Yes.
But you say human can be affected by this disease, but doesn't it like you're taking
care of it?
I'm not sure if we're going to ask the same thing. I was going to ask how different is this TB from the TB that humans have, and if the treatments would be the same or
These are very skilled technicians who should probably be wearing more PPE, but also people who have been studying DTB for a very long time. So they are
how you protect.
like a mask?
yeah, normal PPE, you would wear a mask and gloves if you were dealing directly with body fluids and feces and all of that kind of stuff. The treatment for the 2 TBs is identical.
But the TB is the same.
The species of bacteria is different, but the clinical signs and the treatment are the same. Difficult, expensive, progressing from south to north. So this just shows 2,000, each dot is a herd. So this is a simple yes/no in a whole herd, increasing. It stops around here, then it stops around here, then it's all the way in the north.
Kruger?
Oh, sorry, yeah, this is the Kruger National Park. That's the shaky
Sorry, you're saying that testing's really difficult,
Mm-hmm.
but do you do, even though it's really difficult, you regularly test every herd, and do you cull when you get positive results? Are you doing like kind of farming and things?
No. So the buffaloes are tested routinely for research projects, not by sandparks for management purposes. So for the study that I did, we needed to start with TB-free buffalo. So we collected more than we needed because we knew about half of them would have TB. And then there was a really difficult decision that wasn't in the end made by us, but a foreign ethics committee to release those animals. In previous study, they called the TB positive animals. So yeah, but it's not. There's no culling for TB management.
So a foreign sorry.
Sorry, then they pretty much have to cull everything now.
Yeah.
Yeah, but then so a foreign ethics board can make the decision to cull animals.
Well, it's a registered research project with that university. So their ethics approval came from their university, and it's
they could have
but they have a more conservative ethics board than the local projects.
Okay.
Yes, I think so. It was good.
Yeah. Wasn't it possible to like section off a part of the park to hinder the spread, or rather maybe quarantine the ones that are already thing and then work on those rather than just culling them, testing and culling them?
That approach that you just described is what they did in Botswana. They built a quarantine fence, a massive, long quarantine fence, and the amount of trouble that it introduced in terms of animal migration and the movement of other animals. I mean, this whole western boundary of Kruger is fenced, and that fence is enough to make us realize that it's a lot of management once you put in a fence. Yes.
Are there any prophylactic measures that you take when you find TB negative animals?
There's an active research project to investigate the efficacy of vaccines. They're using the same, one of the vaccines they're testing is the one we get, the BCG vaccine. You have to give it to them when they're very, very, very young. We get ours in our first week of life, right? So they tested in a previous project 3 different vaccines, and the results are not in yet.
Okay.
Even mass vaccination would be a difficult thing to achieve, but they are exploring it.
Okay.
Good. Okay, so what this all means is that there's a strong interest in BTB, and we currently have 18 animals in quarantine bomas in Skukuza. These are TB-free buffalo that we captured for this research project. There's going to be an infection trial, which means we'll challenge them with a massive dose of bovine tuberculosis at the end of July, and the study will look at the disease progression in these animals and various other objectives, but that's the project that's underway anyway. And so this provides us with an opportunity to record the animals before the infection and afterwards, which is fairly unique for a wild ungulate. So, the closest thing we have in terms of baseline data is what I previously mentioned has been collected in livestock and cattle. So there's extensive data on pigs and sheep and horses and chickens. Lots of research out there. Actually relatively little on cattle, and most of it is focused on calves in dairy farms, and they have been using vocalizations to look at those calves. But we do know from cattle that they have these distinct acoustic elements, and that there is information about their age, size, sex, dominant status, reproductive status encoded in their calls and their vocalizations. And that's been quite well. I think I have to speak more quickly. I can summarize the next slide as well. What has not really been studied quite as much is the sounds that they make when they have a respiratory disease. But there are a couple of studies which I'll pull up now that have recorded. Yes. Oh no.
What?
I went too quickly ahead. Okay, so we know the sounds that they make contain information, and we also know a lot about their hearing. So this is the audiogram with cattle in this dash line. They're really sensitive here around 8 kilohertz. So we're going to record their vocalizations. What we're going to do, we can look in that range, provided buffalo have similar vocal characteristics to cattle. So, although bovine TB has not been studied in this environment, other respiratory diseases have, and obviously coughing and sneezing and rails like abnormal breathing sounds are going to be a symptom of advanced TB. And so there's this one looking at calves using an automated continuous monitoring system of cough sounds, and another automatic, and both of these papers are from very recent, like in 2021 and 2022. So this is happening at the same time, which is useful. So the methods in a broad nutshell, we're going to record audio for a few hours a day and video to try and get some idea of the behavioral context of sounds, and also pick up any clinical cough sounds that these animals might be making. And we have a few hypotheses that we might think about. So the very first one is just looking at whether or not they have a similar vocal repertoire to cattle, which will be important in the welfare branch of this research. And then we expect they'll cough more often, there'll be shorter intervals. These are like variables we could analyze. You can pick up the difference in wet coughs and dry coughs. So some coughs might have a totally different behavioral cue, but a wet cough indicates the presence of mucus and disease. And then they found in other animals that there are disease markers in the duration and frequency of calls. I'm not going to ask you guys for ideas because of time. Although maybe I should pause there before we go on to the hippos. So very much a proof of concept study, and hopefully next time I give a talk, I can show you some actual results. Yes, Erin?
I was just wondering about how you would use your sort of like layout for individuals that are positive but completely asymptomatic throughout the entire progression of the disease.
I would say that there's going to be massive variation between individuals and the symptoms that they produce.
Yeah.
Or the only way we can control for it is just we're monitoring the same set of animals at once. Individual localization of the sound might be possible in the same way that I'm going to talk about for the hippos, which would be amazing, but we'd have to get the analyses right.
But in the case they are asymptomatic, you can't really do anything.
Yeah, but
then they don't move, cough or sneeze or anything, right?
But they should make other sounds. It would be difficult. I can also, I don't know. These animals are fitted with, they're fitted with proximity collars and accelerometers, so we do know a bit about the animal's movements as well. But you're totally right. Buffalo take a long time to show clinical signs. In a way, that's part of the question, like how long until we hear the...
Yes. Are these buffaloes going to be held together, or once infected, are they going to be isolated?
So all 18 will be infected. They're held currently in 2 bomas of 9 individuals each.
And they're going to stay in there?
Would you?
Don't you want to get up with several, or
Hmm?
A control, like from those ones.
Well, the control, I think, is temporal, so it's now, before they're infected.
So you're monitoring them before and then after?
Mm.
Yeah.
But
Okay. Would you...
you might have a bias on the weather and everything, right? Because you have the same arrows now. There's another season. They can like behave differently. So it would be nice to have a control together with them to continue monitoring while the time is passing, right?
Yeah.
We're not allowed to make suggestions.
Yeah, no, of course. No, I just think it's impossible at this stage to rearrange the broader study for this control, but you're quite right. I guess the best we can do is monitor temperature and humidity at the same time, so at least we can try and control for season. You can work it into the model, I guess. But you're right.
Would there be any point, I'm assuming there's a lot of data on like viral or bacterial contraction within things, so would there be no point in like infecting some and tracking the what do you call it? The contraction within other members that have not been infected within the...
So they will be sampled every month to look at bacterial load in terms of swabs. So we might be able to use some kind of CFU program, like some kind of measure of bacterial load to look at infection status and so on.
Okay. All right. Because I was just wondering, if you leave some uninfected, you might, this is just like, obviously, blue sky thinking, and that you would have maybe by a fluke one individual or a few individuals that are completely immune and never contract the disease.
Which we will know at the end of the study, because they'll all be
Exposed at some point,
yeah, but also they'll be slaughtered at the end of the study to do all of the lymph node analyses and the histology and all of those fine scale things. So we'll know which animals are sicker than other animals, if that makes sense.
Okay.
Does Zoom also like age-wise for all the 18, they're relatively the same age or are they different?
They're all females and they're all between the age of 2 and 4.
How long do you expect this study to last?
The broader study is quite long term, but this phase will be probably till beginning of next year.
Is there a reason that they're all female?
It's very difficult to keep male buffaloes and bomas. They get very aggressive and they break the bomas and they hurt each other. So with females, they form cohesive herds. I mean, one of the things about buffaloes, they're such social animals, they're so gregarious that they spread TB among themselves very, very quickly. But yeah, the males are just hard to manage. Okay, I'll move quickly through the ideas around the hippo study. So those of you who were in SPAW2023 will know the SPAW2023 golf course, and where we started some preliminary recordings of hippos. And we showed, using these loggers that I'll tell you about, that we can detect simultaneous vocalizations above and below the water using a microphone and a hydrophone. And this, what would you call it, this habitat type, a waterhole with a ton of hippos inside, is replicated quite a few times across southern Kruger. So it's quite an easy place to place loggers when you know the hippos are going to be in amongst those loggers. So what we found is that they seem to primarily use above water communication, but and we know those sounds are much louder and much easier to pick up. But they do make these vocalizations with most of their body under the water, right? So we pick up even above air sounds through the underwater path. And then obviously there's some aerial sounds with no underwater sounds. So we could use that to say the animals are not in the water, they're out on land. So I think we'll get around to it. Yes, okay. So the primary idea is to use a set of 3 or 4 recorders to simultaneously record hippos and use that information to localize them in space. So if they pick up the same sound from the same individual with that many streams of input, we can quite accurately locate hippos in the water. So using the speed of sound in water and the speed of sound in air recorded simultaneously, you'd get a distance to the animal. And with multiple distances to the animal, you can locate them very precisely, unambiguously in space. And we can do the same if the animal's not in our pond, even if it's outside the water, with a little less accuracy, we can still locate it. And so these are the loggers that we're using, these frontier loggers with multiple input channels. So one is the microphone and one is the hydrophone. And so you get your in-air recording, and you can't see it well here, but there's a hydrophone going into the water there. So some of the practicalities that we've run into just trying to get our system to work. Obviously we need a very precise location for each of the loggers if we're going to be looking at the speed of sound as our way of measuring distance. And so these loggers have a built-in GPS, an extremely accurate one. So it locates, I think, 12 satellites, is it 9 or 12? 12 satellites. And using that information, you can very accurately know where each GPS is. And how far away they are from each other is also very important in terms of the calculations. So these were our recording parameters, and this kind of just describes, you guys are more bioacousticians than I am, but you can use these distances and the time that the sound arrives at each microphone to locate your hippo in the water. So I promised Tess that I would run through all of the calibration stuff. So to try and work out, in fact it has a little bit to do with what Adam was saying. We're going to deploy these into a wild situation with all ambient noise and wind and changes in temperature and everything all at once. And so the calibration involved knowing the precise locations of these and making a known sound and measuring the time that each of these sounds arrive. So this is all just to check how precisely we can measure the speed of sound in real world conditions. So very carefully measuring air temperature and humidity, wind speed and direction at the same time so that we can control for those. And so then in the second set of calibrations, put the loggers out far apart from each other and produce a sound right next to each of them, so you know what that sound signal, how that's received in each of the other loggers, and then to calibrate based on that some different points inside our square of loggers. And we tried to replicate something like this in Rondeplay, in the flay there, and we really struggled with the placement of the loggers to get them equidistant apart and to have somewhere secure to tie them. So we have some very preliminary data from that, but the concept works with the loggers, so that's great. So the next step is to deploy the loggers around a place where there are hippos. Obviously, careful placement, we'll know the locations precisely from the GPS, obtain recordings from above and below the water, and then this was some of the stuff we were struggling with in terms of the loggers need to be far enough apart that we can discriminate the differences in the sound arrival time, but not so far away that not all the loggers can actually record the sound. So that was problem number one, and then problem number two was trying to find some way to put them, and it involved a lot of cable ties and duct tape, and an early medium success.
Sharpened metal poles.
So some of the questions we're going to ask using this data when we're finding out some solid hippo data is thinking about the propagation of sound that these hippos make in air and underwater, and think about the ecologically active space of animals in those two different realms. And then thinking about population level behavior, how much communication is going on between one group of hippos, perhaps in the Golf Club Dam, and another group of hippos in Lake Panic just above that, or in the Sabi River, wherever we can find somewhere good to deploy the loggers. It's probably unlikely, definitely in the beginning, that we'll be able to discriminate individual hippos calling unless we have a very controlled environment. Yeah, and then questions around signal structure and source level on land and in water, and how does sound transmission change when you're in the field, in the natural environment with all of these environmental noises going on. That's my talk. Thank you for listening. Any questions about hippo?
Hippos known to vocalize underwater.
Yes. There's very little published work on it, but a few different noises, sounds, vocalizations have been described. Simply recording them is being done by a couple of different people, and trying to get an angle on the behavioral context in which they make those calls can be quite tricky.
Since they spend most of their time in the water, how likely are they to be affected by the BT?
Oh, the hippos. That's a good question. I mean, hippos are, or at least these hippos in Kruger spend most of their nights foraging on land. So they're going to share, and they're grazers, they're bulk grazers, so quite similar to the buffaloes or white rhinos. So they're going to probably share foraging grounds. But to my knowledge, I can't remember if they found BTB in hippos. I have to say, I was thinking about and talking to Guy about attaching animal-born loggers like Adam is doing onto hippos. They are one of the most dangerous animals to immobilize.
I was wondering how you got in that water with. I would never.
I think they haven't been tested frequently for TB just because they're very hard to sample. And probably I can't imagine how many drugs you put into it.
Maybe you could find the TB in the water if you took samples from the water close to those ones in Kukusa.
Yeah, there are quite a few environmental DNA studies of the water sources in Kruger. I haven't checked to see how much they know about TB shedding in more natural water sources. Yeah.
My question is about the recorders. So when you talk about localization, you talk about time of arrival, and that's maybe like milliseconds difference, right?
Yes.
And how much do you guys trust the equipment that you're gonna say that you're gonna start recording at the same time, all of them, and given that it can somehow affect the time of arrival that you combine within all the recorders, you know? For example, if you're using a recorder with the 4 channels, you're gonna trust that's gonna record at the same time. But you work with independently equipment that can behave differently because the machines.
So these loggers set their clock using the satellites. So they should all be well synchronized. I think they should be very well synchronized, or they will each have an extremely accurate clock. And so synchronizing those, we've been using a clicker to make a very sharp sound to be able to align those files. But you're right, our main problem is working in these tiny, these error margins. Yeah, exactly.
But truly, I mean, you cannot start them at the same time, but you need to synchronize them playing the clicker, right? So you know that when this is coming, then you have the same response on all of them, right?
Yeah, I think
Yeah,
that
yeah.
the synchronization is one thing. I think that's quite easy to solve with a satellite clock.
Yeah.
But the errors that you're working with over the distances, especially with the speed of sound in water, you're quite right, you have to be extremely accurate, as accurate as possible. What it means is that we won't know the exact location of each hippo in the beginning while we're refining things, but hopefully it gets easier.
Can you calibrate with a localization?
Yeah, I think, do I not put it in here? It would be ideal to get some...
Like a ground truth?
Yeah,
Yeah.
so I think I put it... Some recordings with known locations of the so we have a rangefinder, and if I stand right at the position of the logger and use a rangefinder, we can at least get maybe a few calls where we know the position of
It was fairly good in Skukusa because you could actually see them.
You can see, yeah.
So this was rather terrible in Ronderplay because we couldn't see any of these hippos, but in Skukusa you could see them.
Yeah. Any last questions? Thank you for staying so late.